Stefano. Yeah, so I'll be doing a little bit of an overview of the projects we've been working on, like Stefano said, at Radboud with Joe. So my title is Multiomic Analysis of Aniridia Stem Cell Derived Corneal Cornea Cells. So you all know the cornea is, so we all know that the cornea is the transparent layer at the very outside of the eye. And an important part is the limbus, which sits just right outside as a ring that contains this limbal epithelial stem cell population. And these cells are very important because they are the ones who continuously replenish the central cornea epithelium. Without them, we all know there's limbal stem cell deficiency, and we also all know that aniridia patients suffer particularly from this disorder. So it's very important for us to understand how PAC6 regulates this limbal stem cell function and how the process occurs, so that we try to identify targets that can either prevent aniridia-related keratopathy or at least treat it. No, this is back. Okay, so what are omics technologies? So omics are these very recent high-throughput technologies that allow us to study the cells as a whole. So for example, in transcriptomics, where we look at gene expression, we don't just look at one gene expression, we can really study all the genes that are in these cells. And for this, there's two major techniques. One is called bulk RNA-seq or single-cell RNA-seq. So bulk, this is the best analogy I could find. So think of bulk technologies or bulk RNA-seq as a smoothie. So we have a taste of what's in there. We can tell, what kind of fruits are in there, but we cannot really tell exactly, for example, the smaller population. So, for example, if we just add a couple of blueberries in there, we might not even taste them, so we might not know they're there. As for single cell, think of a fruit bowl, where actually we can see exactly all the cell types that are in there, how much are there, so even the smaller populations we can detect. And then, I keep going back, okay. And then we have epigenomics, so another omics technique that looks at how these genes are regulated. So it detects before the gene is expressed how this process works. And this is a bit complex, but I hope you can follow me, but because I really need to explain you right. So basically what we have in the chromosome, we have all the DNA, which is this string here in blue. And when the gene is turned off and is not really active, the DNA parts are tightly bound and tightly wrapped in these complexes with these orange proteins called histones. And then when the gene is necessary and it needs to be expressed in a particular cell, this complex loosens, and then the DNA becomes available. And you see here in red. And this is where the transcription factors, which are the proteins that turn on and off the genes, and PAC6 is a big important one, can bind and then turn on the gene. So this technique called ATAC-seq can actually detect and sequence all these regions that are open, and we can have an information on that. So, as cell models in our lab, we use induced pluripotent stem cells. I think I've shown you this before, but we can generate induced pluripotent stem cells, or iPSCs, from a patient sample. So for example, a skin sample. We extract the cells, and then we give them a cocktail to reprogram them and make them go kind of backwards so that they become more pluripotent and undifferentiated. And then from them, we can actually have a source of cells that we can differentiate into the very different cell types. So we can do cornea cells, we can do retinal cells. In theory, we can do any type of cell because these cells have that potential. And then, of course, having cells that have the genetic information as the patient, we have an amazing tool to study disease, for drug screening, to treat the patient, and of course for transplantation. So these cells have a huge potential. Yeah, and like I said, they're a valuable source of cells that have the mutation. So going into the first technique that we use, so we did bulk RNA-seq. This project actually started when I was still at Mariya Moosajee's lab at UCL. And what we did was we had 4 lines. We had 2 control lines, so they don't have the PAC6 mutation. And we have 2 iPS lines generated from 2 aniridia patients. Mutations are here, so they're both nonsense mutations. and we differentiated the iPSCs into limbal epithelial stem cells. And I just call them ILSC because they come from the IPS, so we call them ILSC. This process takes about 25 days, so we picked samples at day 0, where there's still IPS, at day 15, so more or less in the middle, and then at the end point. And we did the bulk RNA-seq, so think of smoothie in these next slides. And this is what an overview of what we get. I will go through it with you. So this graph shows how separate the samples are, how different they are. So if they are very different, and especially along this axis is where most of the variance is. So cells that cluster here and here, for example, they're very different. Cells that cluster together, they are more similar. So their gene expression profiles, the whole gene expression profile is more similar. So day 0 iPSCs, they cluster all there together as we expected and quite far apart from everything else because they are indeed quite different. We added a positive control, which are actually limbal stem cells extracted from the cornea. We had our day 15 samples. These are all the triangles up there. They stayed very close. So aniridia and wild type, this tells us they're not majorly different. And then the day 25, where we actually see quite a separation. So you can tell all the day 25 controls are here. They sit together. They're still a little bit far from the primary limbal, which is normal. They're still a bit immature compared to the actual cells from the tissue. And then the day 25 sit here. And actually, there's also some separation between the two lines. And I'll get back to this later. So we look deeper into this, what kind of genes are expressed at this time point. We see that for the control, we have 400 genes that are more expressed in the control samples than the aniridia. And when we look at what these genes do, they're all related to processes that are quite important for the cornea. So we see epidermis skin development. This is because the skin epithelium and the cornea epithelium is quite similar genetically. So the processes, so this tool actually only gives us skin related because there's not a lot of cornea information in there. But trust me, this is important for the cornea. We have cell adhesion, extracellular matrix organization, and wound healing. And then we also looked at what genes were up on the aniridia compared to the control. And this is a bit surprising. We saw cell division, replication, and neuronal cells. So it feels like these cells express more neuronal genes than they should. But I digged a bit deeper into the genes that are downregulated in aniridia, and I went to fish the specific limbal stem cell markers because I wanted to see how they look. And we see an overall downregulation of the limbal epithelial stem cell genes. So if you see here, every line is a different gene, and every column is a sample. So all the wild type or the control samples sit here, and they have red values because the gene expression is higher. And then if you look at all the aniridia there, they are all blue, which means that all these genes are downregulated. So there's really a sharp difference in the terms of the limbal epithelial stem cell genes. So because we see this variability between the two lines, we are a bit worried that what does this mean? Do these results are because of PAC6 itself, or are there something else going on? And the best way to solve this question is to do isogenic lines. So we pick the aniridia 1 and aniridia 2 lines, we correct each of the mutations, and then we have an exact control for that line. So we're doing that at the moment. For the aniridia 2, it feels promising. I already have 2 clones that have the mutation corrected, and they're growing nice, so I'm hoping they'll be validated soon. Aniridia 1 hasn't worked yet, so I'm still working on it. But then hopefully when we have these lines, we can differentiate them as well. We can take RNA-seq samples, and then we can really answer if the results back in the last slide are indeed because of PAC6. And then we can answer these questions. Can we rescue these changes by correcting the mutations? How really can lower PAC6 affect this process and affect limbal stem cell development? And can we find actually target genes or pathways like it was shown before for therapy development? So then the second study, now we're going into the fruit bowl. We did the same process, so exactly the same, or pretty much the same protocol, We picked 3 time points. This time we actually worked with Scottman Lab from Tampere. They are the ones who did all this iPS differentiation work. And then we collected these samples and we sent them for single-cell RNA-seq and ATAC-seq. And this is a little overview. The paper is actually pre-accepted. We're still waiting for the final decision, but it's looking good. And then what we see is, again, a general graph with all the samples plotted. And this time, every dot here is a cell. And we can see that there's a good separation between the different cell clusters. It's what we call the little, where the cells clump together, we call them clusters. So we see day 0 there, clustering again separate. We see the middle time point, day 11 here, and we see the day 24, the final time point there. What we also saw at day 24 was that not all cells group together. We see these little 2 populations, so 9 and 8, that are also present at this time point, but they really seem to be quite different. We then looked at what all these clusters were based on their gene expression. We managed to identify all of them. I don't go into detail here, but I can tell you that cluster 5 up there is the one that mimics the most the limbal epithelial stem cells. So that's our target. That's the one we wanted to achieve. What we also saw is that when looking at each individual cell line that we used in this study, we see variability in the production of these cells. So we have an ESC line, embryonic stem cell line, that actually is very efficient at producing limbal stem cells. We see 95% of the cells at the last time point are clustering in here. But for the iPSC lines, we see lower percentage and variable, so between 22 and 35. So that makes us think, how can we actually tweak this protocol to get more ILSCs from these lines? So we looked into the RNA-seq data again, and we've looked for surface markers that were expressed just in that limbal stem cell cluster, and we found 2, AREG and integrin alpha 6, and we did FACS sorting. So we fished only the cells that express these markers, and that was actually quite nice, because if you see here, after we select for these cells, we see in the bottom here is the selected, and here is the not selected. So we see epithelial morphology improved quite a lot. We see we have stronger signaling from a lot of the limbal stem cell markers, and luckily, or not luckily because it was a lot of work, but importantly, we used to see this weird population that I showed in the back that still had a bit of a iPSC identity. We got rid of it because we don't see any pluripotent stem cell markers anymore. So this strategy really improved not just the limbal stem cell production, but the consistency. And this is really important for transplantation, for example, or stem cell-based therapies. And then for the last study, for this ATAC chromatin study, so this is still really just very preliminary. It's really the first analysis we're doing. We see comparable results to the single-cell RNA-seq, and I will show you a little bit. So again, we have the general graph where with all the dots which are a cell. We see again some separation. So day 0 stand right there, day 11 here, and day 25 here. And in here, we could identify 6 clusters, not 8. So again, there's a little difference that we're still looking into, but we see 6. And each color here in the graph represents a cluster. What we could already identify is that the limbal stem cell cluster is the green one over there. And in this case, it's about 50% of all the cells that show this ILSC. So we're still identifying the other clusters now. One thing that I wanted to show here is because we're looking at chromatin openness, right? So we're looking at the openness of the gene body. I looked at PAC6 gene body. PAC6 is always a sketchy, it's always complicated no matter what. What we actually see here, the color means, so the more open the gene body is, the brighter the color. So if you look at yellow, it's where it's most open. And actually what we see, even though it feels a bit open everywhere, or nearly everywhere, the highest is on day 0. So that we didn't expect at all. Although we know the PAC6 gets turned on very early in this process, but we still didn't expect that the highest would be on day 0. So if anyone has any suggestions why, I'm happy to hear it. But we can also look at all the other markers that we want to, the limbal stem cell related. So we look at these two. P63 is a very important transcription factor as well, that we also want to look into processes related to that. And we see it's really bright yellow in the limbal epithelial here. And then we're now looking at keratins, which are also important markers for limbal stem cells, and also for the bottom ones, keratin 3 and keratin 12, are direct targets of PAX6, so we can look at those as well. But although keratin 3 is already a little bit turned open here, keratin 12, which is a later corneal epithelial marker, indeed, we cannot detect it. So it feels like it's still closed, so it should be not expressed. But this is just an overview of what I wanted to show. We still haven't applied these to Aniridia iPSC lines yet. We are doing it at the moment. But just a quick summary on how we could use these for Aniridia research. So we know that single-cell RNA-seq, it identifies all the different types of cells, and we can track how they change over time. And we, for example, I showed, how do I say? Yeah, we show that it improves differentiation efficiency, for example, for cell therapy potential, but it has a lot more other purposes. For the ATAC-seq, we see it measures openness of chromatin in each cell. We can understand with this technique how this gene expression in limbal epithelial stem cells occurs, and importantly, how it can be regulated by PAX6. So I really hope next time I can show you that. And not just by PAX6, by any other transcription factor. And importantly, we can actually combine both data from both techniques and do a data integration step, where then we can really see and identify all the regulatory networks. And then I can also show you a complicated graph full of arrows that can be regulated by PAX6. And this will be really important to understand how how PAX6 works in this process, and try really to find new targets. And then lastly, I just want to say that we are upping up a notch, and now instead of doing just one cell type, we are going to do organoids, and I'm already starting with that. You can see a beautiful transparent bubble here. And with this, we want to see how PAX6 affects the full cornea development. So not just at the epithelial part, but really the stroma and the endothelium. And yeah, I hope at the next meeting I can show you some more of this. Yeah, and I would like to acknowledge a lot of people who have contributed for this work. So mostly people from the Radboud, from Joe and everyone in the group, Mariya and the group at UCL, where I started all this Aniridia adventure, and Heli Scottman and the group at Tampere University. And thank you very much.